Hello all. Now we will discuss something about the metabolism of carbohydrates. We will discuss few important reactions in a very simple way regarding some of the important metabolic reactions of carbohydrates. So first thing is about the glycogenesis. Glycogenesis, it is the synthesis of glycogen from glucose in the liver and muscles is called as glycogenesis. So, the various important reactions which are occurring here in the glycogenesis. The first one, the glucose is converted into glucose 6 phosphate in the presence of the enzyme hexokinase and glucokinase. Then this glucose 6 phosphate is converted into glucose 1 phosphate in the presence of the enzyme phosphoglucomutase, and then this glucose 1 phosphate combines with uridine triphosphate in the presence of the uridine diphosphate glucose pyrophosphorylase, and then it is converted into uridine diphosphate glucose. This uridine diphosphate glucose, it transfers its glucose to non-reducing end of a pre-existing polysaccharide primer, and in the presence of the enzyme glycogen synthetase, it is converted into alpha 1,4 glycosyl units. This alpha 1,4 glycosyl units, in the presence of the enzyme, branching enzyme, is converted into a branch of glucose, that is glycogen. So this is about the glycogenesis. The next thing is we will discuss about the glycogenolysis. Glycogenolysis, it is the breakdown of glycogen to glucose in the liver and kidney, is called as glycogenolysis. Here in glycogenolysis, glycogen is converted into glucose 1 phosphate in the presence of the enzyme phosphorylase. Then this glucose 1 phosphate is converted into glucose 6 phosphate in the presence of the enzyme phosphoglucomutase and in the presence of magnesium. This glucose 6 phosphate is again converted into glucose in the presence of the enzyme glucose 6 phosphate. So this is about the glycogenolysis. The next thing is about the glycolysis, or it is also called as Embeden-Meyerhof pathway. The conversion of glucose or glycogen to pyruvate and lactate is known as glycolysis. Lactate is produced from pyruvate only under anaerobic conditions. Under aerobic conditions, pyruvate enters the Krebs cycle through acetyl CoA for further oxidation of glucose. Now we will see the various reactions which are occurring there in the glycolysis. First, the glucose is converted into glucose 6 phosphate with the help of enzyme hexokinase. Then this glucose 6 phosphate is converted to fructose 6 phosphate by the enzyme phosphoglucose isomerase. Then this fructose 6 phosphate is converted to fructose 1,6 diphosphate in the presence of the enzyme phosphofructokinase. Then fructose 1,6 phosphate fructose 1,6 diphosphate is converted to glyceraldehyde 3 phosphate with the help of enzyme aldolase. Some amount of fructose 1,6 diphosphate is also converted to dihydroxyacetone phosphate by the enzyme aldolase, which is again converted to glyceraldehyde 3 phosphate by the enzyme triose phosphate isomerase. Glyceraldehyde 3-phosphate is converted to 1,3-diphosphoglycerate with the help of enzyme glyceraldehyde phosphate dehydrogenase, and then 1,3-diphosphoglycerate is converted to 3-phosphoglycerate with the help of enzyme phosphoglycerate kinase. 3-phosphoglycerate is converted to 2-phosphoglycerate with the help of enzyme phosphoglyceromutase, and 2-phosphoglycerate undergoes dehydration and converted to phosphoenol pyruvate with the help of enzyme enolase. Phosphoenol pyruvate is converted to pyruvate by the enzyme pyruvate kinase. Pyruvate is converted to lactate under anaerobic conditions, and pyruvate enters the Krebs cycle under aerobic conditions through acetyl CoA. So this is about the Embeden-Meyerhof pathway or glycolysis. The next thing is about the citric acid cycle, or it is also called as Krebs cycle or tricarboxylic acid cycle, that is TCA cycle. The oxidation of pyruvate to carbon dioxide and water under aerobic conditions is called as Krebs cycle. So now we will see the various reactions which are occurring here in the Krebs cycle. So first, the pyruvate which is generated during the process of glycolysis will enter into the Krebs cycle. So this pyruvate undergoes decarboxylation reaction with the help of pyruvate dehydrogenase complex to form acetyl CoA. This acetyl CoA combines with oxaloacetate to form citrate. This reaction is catalyzed by the enzyme citrate synthetase. Then this citrate is converted into cis-aconitate, or also known as isocitrate, with the help of the enzyme aconitase. Then isocitrate is converted to D-isocitrate or oxalosuccinate with the help of the enzyme aconitase. Then this D-isocitrate is converted into alpha ketoglutarate with the help of enzyme isocitrate dehydrogenase. Then this alpha ketoglutarate with the help of enzyme alpha ketoglutarate dehydrogenase is converted into succinyl CoA. Then this succinyl CoA is converted to succinate, and this reaction is catalyzed by the enzyme succinyl CoA synthetase. Succinate is converted into fumarate with the help of enzyme succinic dehydrogenase. Fumarate with the help of enzyme fumarase is converted into malate. Malate is converted to oxaloacetate with the help of enzyme malate dehydrogenase, and thus the cycle continues. So this is about the citric acid cycle or Krebs cycle. Then next thing is about the gluconeogenesis. The formation of glucose from non-carbohydrate sources is called as gluconeogenesis. The richest non-carbohydrate sources are lactic acid, amino acid, and glycerol. When the carbohydrate reserve in the body is exhausted, normal blood glucose level is maintained by glucose, which is formed by this process. The richest non-carbohydrate source is lactate. This lactate is converted to glycogen in the liver. Glycogen is converted to glucose and enters into the blood. And this glucose re-enters into muscle in the form of glycogen. This is called as Cori cycle. Here you can see this is the Cori cycle. Now the various reactions which are occurring there in the gluconeogenesis. So the richest source of gluconeogenesis is lactate. Then other than this, the other sources are amino acids and glycerol. The lactate as well as amino acids, they are converted into pyruvate, and this pyruvate is converted into oxaloacetate, then it is oxaloacetate is converted into phosphoenol pyruvate is converted to 2 phosphoglycerate, 2 phosphoglycerate is converted into 3 phosphoglycerate, 3 phosphoglycerate is converted into 1,3-diphosphoglycerate, and 1,3-diphosphoglycerate is finally converted into glyceraldehyde 3-phosphate. And the glycerol which is there here, this glycerol is also a source of non-carbohydrate source for carbohydrates. So this glycerol is converted into dihydroxyacetone phosphate with the help of the enzyme glycerol phosphate dehydrogenase. So this dihydroxyacetone phosphate, it combines with glyceraldehyde 3 phosphate to form fructose 1,6 diphosphate. So this fructose 1,6 diphosphate is converted into fructose 6 phosphate, and this fructose 6 phosphate is finally converted into glucose 6 phosphate here. It is converted into glucose 6 phosphate, and this glucose 6 phosphate is finally converted into glucose. So this reaction is known as gluconeogenesis. So these are some of the important metabolic reactions of carbohydrates. Thank you.